Welcome back. In this part, I'll explain about the different stages of mitosis as a part of cytology practicals. Mitosis is an equational division. It occurs in all the somatic cells, and a parent cell will divide into two daughter cells, and both the daughter cells will receive the same number of chromosomes and same amount of DNA without any alteration. In order to understand mitosis, we divide it into different phases: interphase, prophase, metaphase, anaphase and telophase. I will tell you briefly only for the sake of practicals. The details you will get it in the theory class. The phase between one cell division and the other cell division is interphase. The moment the cell decides to divide into two daughter cells, it enters into prophase. In prophase, the chromosomes appear in the form of thin threads. We call them chromatin. Nuclear membrane is visible, nucleolus also is visible, and the genetic material starts condensing. Condensation begins in the prophase. That is what is prophase. Prophase is very elaborate in meiosis. The next phase is called metaphase. Metaphase is the most important phase in case of mitosis because the chromosomes are condensed to the maximum extent and they are present on the equatorial plate or metaphase plate or in the middle of the cell. So the next stage is called anaphase. Anaphase is the phase where the chromosomes which are duplicated, which have undergone replication, one chromosome has become two. These two chromosomes will now start separating from one another. Now the chromosomes are now moving towards the opposite pole, and this is called anaphase. Telophase is the phase where the chromosomes have already reached the opposite pole and they start forming their own nucleus. So thus two daughter nucleus start forming, and before that telophase is found. So very distinctly, prophase is the phase where the chromatin is seen in a decondensed form. It is only in the form of thin threads. We call it as chromatin. Metaphase is the phase where the chromosomes have condensed to the maximum extent and the chromosomes are found on the equatorial plate or the metaphase plate. And anaphase is the phase where the chromosomes start moving away towards the opposite poles. And telophase is the phase where the chromosomes have already reached the opposite pole and start forming their own nucleus. And after telophase, cytokinesis occurs. In the middle of the cell, wherever there was a metaphase plate, the vesicles start fusing and they form the cell plate. So as a result of the formation of a cell plate, two daughter cells are produced. This is a brief account of mitosis. You will study details in the theory class. This is only for the purpose of practicals. Welcome back. In this part, let us study the different stages of mitosis. These are some of the important points related to mitosis. It was actually Strasberger in 1875 who studied mitosis for the first time. The continuation of species from one generation to the next generation is governed by two important processes. One is called syngamy, which is union of gametes. The other one is cell division, which is actually marked by mitosis and meiosis. Anaphase and metaphase in both mitosis and meiosis are very, very important. The duplication of DNA, that is replication, occurs in the S phase, which is actually a part of interphase. There is endomitosis, where there is duplication of chromosomes without cell division, which normally doesn't occur in nature. It can be induced by some chemicals like colchicine. This is Edward Adolph Strasberger and his original drawings when he first observed mitosis. Interphase is the period between one cell division and the other cell division. In mitosis, a single nucleus gives rise to two identical nuclei and also two daughter cells. During metaphase, the chromosomes with kinetochore arrive at the equatorial plate. So metaphase is very important in mitosis. Anaphase begins when the centromeres separate and chromosomes start moving from the metaphase to opposite poles. When chromosomes complete the movement, then telophase begins. Nuclear envelope and nucleoli will actually reform. Cytokinesis, which is the last stage of mitosis, occurs by the fusion of vesicles to form a new plasma membrane, which is a cell plate is formed between the two daughter nuclei. So that is the end of mitosis. These are the diagrams that represent mitosis. We have the prophase here with a nuclear membrane. The genetic material is in the form of chromatin yet. This is the early metaphase. The chromosomes are now moving towards the equatorial plate. The chromosomes are condensed. You can see them as distinct structures. This is metaphase. All the chromosomes are there at the metaphase plate, and the centromeres of the chromosomes are attached to spindle fibers. This is anaphase, where the chromosomes have already been separated. These are called sister chromatids. These chromosomes are separated from one another. These two, which are actually copies of a same chromosome, will now separate and move towards the opposite pole, and they are actually pulled by the spindle fiber. This is telophase, where the mitosis is already come to an end, and this is where the cell plate is produced. The cell plate is formed by the fusion of vesicles, and these are the two new nuclei that are formed. This is the diagrammatic representation. Actually, these are the actual pictures of mitosis. This interphase in the low power, interphase in high power, prophase in low power, prophase in high power. This is metaphase in low power, metaphase in high power. You can see all the chromosomes aligned on the equatorial plate. Anaphase in low power, anaphase in high power. One more slide to show you the diagrammatic representation of different stages of mitosis. Interphase, where the cell is mostly in interphase. The moment the cell decides to enter into, the moment the cell decides to undergo mitosis, it enters into prophase. And in the interphase there is S phase. In S phase the duplication of chromosomes occur. So by the time it comes to prophase, already chromosomes are multiplied. By the time it comes to prophase, already chromosomes are duplicated, but they are appearing in the form of thin threads which are called chromatin, and you can see the nuclear membrane still, and you can also see the nucleolus. Metaphase, where the chromosomes already are aligned at the metaphase plate with centromere at the metaphase plate, and each chromosome is attached to spindle fibers. The distinct difference between the metaphase of plants and animals is there is no centriole formed in plant cell, whereas centriole is formed in the animal cell. This is anaphase, where the chromosomes already reached the opposite pole, they are moving towards the opposite pole. This is telophase, where chromosomes have already reached the opposite pole, they have already started forming their own nuclei, and cell plate is almost formed by the fusion of vesicles. One more difference between plant cell and animal cell is plant cells appear rectangular, animal cells will appear circular. That is another difference between plant mitosis and animal mitosis. This is the jiffy to show you the formation of cell plate by the fusion of vesicles. You can see the cell plate is formed here. This is only a diagrammatic representation, and this is also representing the cytokinesis. That is the brief account of mitosis. Thank you very much for watching this video. Please like, share and subscribe. We will meet again in the next video with some other topic.